From the West Coast, and is Dr. Andrew Whitehead, who is currently an associate professor at UC Davis, and a member of the population biology group and Coastal and Marine Sciences Institute. Dr. Whitehead obtained his bachelor's from the University of Guelph in Ontario, and then went on to do his doctoral work under the direction of Susan Anderson at UC Davis, and then went on to do postdoctoral work at the University of Miami Rosenthal School of Marine and Atmospheric Science. His work, he's most noted for studying the killifish and its ability to acclimate and adapt to really inhospitable conditions most organisms wouldn't be in, such as ecotoxicity in the Gulf, as well as its ability to live outside of water, the mangrove killifish. And I think many in Carnegie would be interested to know he also works on osmotic stress tolerance in the red abalone as a predictor for ocean acidification and the ability of these organisms to not only evolve and adapt to the changes in the pH of the ocean, but to use it as a predictor for how organisms can evolve and survive in the ocean. Today he's going to tell us about his work on the killifish, and his title is Evolution in the Anthropocene, the genetic basis of rapid and repeated adaptation to environmental pollution in fish from urban estuaries. Thank you.
Great. Thank you, Tyler, and thank you to the organizers for putting together such a really rich and diverse meeting. I've certainly learned a lot. It's great to go to meetings that are as diverse as this because you get really yanked out of your comfort zone and learn lots of new things. So it's an honor to be here. Thank you so much for the invitation. So humans have a pretty large global footprint. We're moving pretty big levers on a planetary scale. I think the term Anthropocene was perhaps controversial in its usage maybe 15 or 20 years ago, but I think most people would agree now that this isn't a controversial term, that humans are affecting landscapes at a global scale. And so we think of the big levers, we're thinking of things like climate change, harvest pressure, or environmental pollution. And when you think of the long-term consequences of this for wild species, there are a number of, so at least at a course level, a number of alternatives for how species may respond to this kind of environmental change. Either move away from where this pressure is, adjust your physiology to the change in the environment, individuals through phenotypic plasticity that we've seen some wonderful examples of just in the last little bit. Another option is to evolve, a phenomenon that emerges across generations, so beyond the lifetime of individuals at which plasticity emerges. Or, of course, it's also an option to suffer and go extinct. So we're interested both in acclimation responses and in evolutionary responses to environmental change, and we grapple with issues associated with climate change and pollution, acclimation and adaptive responses. But today I'm going to talk about evolutionary responses to chemical changes in the environment from contemporary pollutants. I like taking a long view on this because the contemporary, we can think of sort of chemical changes in the environment as a very contemporary phenomenon, but of course there have been radical changes to the chemical environment over the span of life, and there have been evolutionary responses to these changes in the environment, of course. So there's a long history of adaptation to environmental toxicants. So we think of challenges probably in the very earliest life forms was being able to deal with the damaging effects of UV radiation, right? So of course now we have an ozone layer, but the earliest life forms didn't have an ozone. The earliest life forms made the ozone layer, right? And so those earliest life forms needed to be able to deal with the damaging effects of UV radiation. Of course, once photosynthesis really got up and running, then there was another radical change at a planetary level in terms of the oxygenation of the atmosphere. Oxygen's a pretty toxic molecule in many ways. It's really damaging to lots of cellular structures through the generation of oxygen-free radicals. So probably, these big boosts in oxygenation of the atmosphere probably coincided with some of the first mass extinctions. And there's no evidence of that, or no record of that in the fossil record, because bacteria didn't fossilize for the most part. And also with the oxidation of the atmosphere, you've got pretty big changes in the bioavailability of metals. So things like iron, that was essential for life, all of a sudden becomes less bioavailable in oceans with oxygenation of the atmosphere. And other metals that were pretty toxic and nasty all of a sudden become more bioavailable in oxygenated environments. So these are pretty radical changes in the environment that life had to evolve in response to. And then, of course, we've got chemical arms races between plants and herbivores are some of the best examples. And we've got plants that are recruiting metabolites that they're able to use as defensive molecules. And then, of course, animals, herbivores needing to evolve mechanisms to be able to metabolize and deal with these noxious and toxic chemicals. Things like ricin and castor bean, made famous by Walter White. Everyone knows about ricin now. But atropine, nightshade berries, and various toxins produced by plants. And of course, there have been adaptive solutions to all of these kinds of challenges. So we think of the evolution of pigments and DNA repair systems. So DNA photolyase is an ancient enzyme system that's shared by all life forms. So that evolved very, very early. Mechanisms that deal with oxygen toxicity and mechanisms that deal with the sequestering and pumping and mobilization of metals are also ancient. That date to organisms evolving in this time period. And of course, when life emerged onto land and the arms race between plants and herbivores really took off, we get this huge diversification of phase 1 and phase 2 metabolism systems like the cytochrome P450s and such. Okay? So, and these defense systems, the DNA repair systems, the metallothioneins, and the cytochrome P450s, for example, are the same sort of metabolic systems that are called upon in contemporary organisms to grapple with the chemicals that we're putting in the environment. So of course, the difference with contemporary environments is that this chemical environment is perhaps changing too quickly for certain species to be able to keep up. And so it's not like life hasn't evolved continuously to all these chemical changes in the environment. It's not like life can't evolve in response to these things. It's just that some species may be limited in how quickly they can evolve. But these evolutionary histories matter for the here and now. These defensive systems are called upon. And they also can contribute to understanding species variation and how some species may be more or less sensitive. So if you think, there are different species of frogs, some lay their eggs near the surface that are exposed to UV. Other species lay their eggs at depth and in the shade. And those species that lay their eggs at the surface have fairly robust DNA repair systems and are more resistant to the DNA damaging effects of some of the chemicals that we're putting into the environment. So some of that evolutionary history really matters and can help us think about how some species may be more or less sensitive to the chemicals that we're contributing now. So when we think of evolutionary toxicology, there's this sort of tragic irony that I like to think of in contemporary toxicology. The species that we intend to murder with chemicals are the ones that tend to evolve adaptations and resistance to these chemicals. And the ones that we don't intend to kill with these chemicals are the ones that really tend to suffer the consequences of deploying these chemicals into the environment. So, we like to think about what are the attributes of species and what are the attributes of environmental change that may enable or disable species' ability to evolve resistance. So the ability for evolution to rescue a species from a radical environmental change depends on many things. It depends on the properties of the environment, of course. What's the magnitude of change in the environment? And what's the rate of that change in the environment? And what's the availability of alternate habitats to be able to hide out in? The likelihood of evolutionary rescue also depends on properties of the organism, their natural history, their population sizes, their ability to move away, their generation time, these kinds of things. And also it depends on the properties of the traits that are under selection, or the traits that may be adaptive to these altered environments. Is there genetic variation that pre-exists within populations for these traits? What's the potential for phenotypic plasticity to be able to adjust at a physiological time scale that might release you from selective pressure? And also the traits, if there is genetic variation, how many genes are involved? What are the interactions between those genes? All contribute to how we may think about and estimate how species may be able to evolve quickly. And I would argue that there are additional considerations when we think about chemical toxicants, in addition to some of these here. One is the complexity of the chemical mixture that you're exposed to might contribute to the complexity of the adaptive phenotype that's necessary to survive in that environment. So we know that a lot of pollution is encountered as a complex mixture of chemicals. Whereas a lot of the adaptive scenarios that we can imagine to poisons, like adaptations to pesticides and antibiotics, are typically adaptations to fairly simple chemical exposures. You're exposed to maybe one or a small set of chemicals. And many of those chemicals where we've seen adaptive evolution, usually unwanted adaptive evolution in terms of pesticide resistance, are in response to chemicals that were designed to kill. They were designed as poisons. And I think that matters because when we design chemicals to kill, like antibiotics and pesticides, we're designing chemicals so as they don't kill everything. We want them to kill just the things that are a pest or are causing disease. So they're designed to have very specific molecular targets, right? And so perhaps you just need a few changes in the molecular target to evolve resistance. Whereas a lot of pollutants, chemicals that weren't designed for their poisonous or lethal properties, are still poisonous and lethal in many scenarios, but might have many different molecular targets that contribute to a toxic or disease outcome. So perhaps there are many more genes that might need to change to contribute to resistance when you're exposed to not only complex mixtures of chemicals with different mechanisms of action, but for each of those chemicals, there's maybe a complex mechanism of toxicity that you need to evolve your way out of. So again, these critters have very rapid generation times and huge population sizes and are exposed to simple mixtures of chemicals typically and can evolve quickly, whereas these critters are typically the opposite of what I just said. Okay, so we've been working on a critter that perhaps, phylogenetically, you would lump in with these guys and life history-wise, and that's this cute little guy here. So this is Atlantic killifish. This is our charismatic biofauna, perhaps. They're about this big. They're pretty cute, right? So they live in pretty dynamic environments to begin with. They're estuary specialists, and estuaries are pretty hard places to make a living, right? Because they're environments that are changing on multiple times a day with the coming in and going out of the tide and seasonal. So you got big changes in depth, big changes in temperature on a daily scale, big changes in salinity with the tide going in and out, big changes in respiration day to night. So CO2 is changing, acidification is changing, nutrients are changing. So it's a pretty dynamic environment, and these guys are amazing survivors there. So if you go to the East Coast, so you just go out to the coast from here in Chesapeake Bay, and you throw in a minnow trap, or you drag a seine net through these environments, these guys make up almost everything that you're going to catch with a backbone. So there's very, very large population sizes in these salt marshes. They're very successful. They can go, they can acclimate to fresh water all the way up to 4 times the salinity of seawater. Like the salt's falling out of solution before you can kill them. So they got this huge dynamic range there. Of course, they don't mind lots of oxygen, and when you start, slowly purging the oxygen out of the water, they'll just, once it's all gone, they'll just come up to the surface and start gulping air. They span a range from Nova Scotia down to Florida, so it's a pretty wide temperature range. So pretty tough little guys. But we also find them in these really radically transformed environments, environments that have been transformed, evolutionarily speaking, very, very recently. Obviously, there's a lot of habitat loss in human-altered environments along the coast. But even where there is suitable habitat, like salt marsh, it's also pretty altered on a chemical scale. There's a lot of persistent organic pollutants in a lot of these sites, including also heavy metals and other things. Okay? So here, there are lots of dynamic natural stressors, and we use them as a model system to understand some of the physiology and genetics and molecular biology that underlies this amazing physiological flexibility. So we're using it as a model to study sort of physiological flexibility or phenotypic plasticity. But in these environments, this has been sort of a permanent alteration of the environment, this chemical change in the environment. These are very, very persistent chemicals that have been there for decades and are going to be there for centuries. So here's a permanent alteration or deformation of the landscape that's not sort of flexing in and out. It's not a dynamic environment. It's this permanently altered environment. And here we see the evolution of locally specialized types that can actually survive in this pretty altered environment. So when we think of killifish, people are like, oh, well, of course they've evolved resistance to pollutants. They're such tough little fish. It turns out that they're tough when it comes to natural stressors, but when it comes to chemicals, they're total sissies. So here's a bunch of different species of fish, right? So trout to zebrafish and herring, catfish, minnows, what have you. And here's a model sort of toxicant, dioxin, and this is the amount of chemical it takes to kill you, right? And so killifish are down here with trout. I mean, total sissies. They can't tolerate all that much chemical. Many classes of chemicals, they're like this. So it's not like they're very resistant to begin with compared to other species. However, we study populations along the East Coast that people found in the 1990s that all 4 of these sites, these bolded sites here, are Superfund sites. They're sites that have been designated by the EPA as the most horribly polluted sites in the nation that have been prioritized for cleanup. And there are normal, healthy-looking killifish populations there. And it's not like Blinky the fish with 3 eyes. These are normal-looking fish, right? So we've been studying these 4 polluted site populations. And for each of these polluted site populations, we study a geographically nearby, clean site population. So we got these 4 pairs of clean polluted site populations as you go from Massachusetts down to Virginia. So this is just a sailboat ride down the Chesapeake Bay. So when we bring these fish from these populations in the lab, keep them in the lab for a couple generations so we can compare heritable differences between them. And these are the survivorship curves of our reference site populations when exposed to a model toxicant. Here it's a PCB, increasing log doses of PCB 126. And we can see, they all die at a reasonable concentration. But when you look at the fish from the polluted sites, they're really extraordinarily resistant. I should remind you that this is a log scale here, so this isn't a subtle difference in resistance, right? So it's a pretty extreme tolerance phenotype. So we go back to here. These are a normal killifish. If we're to superimpose our polluted site killifish, this is their level of resistance. Right? Jaws are hitting the floor. That's ridiculous. And this is a log scale, right? So this isn't a subtle twofold difference in some phenotype. These fish are able to resist thousands of times the normally lethal concentration of these chemicals. And that's evolved by natural selection in a few dozen generations. So within a single species, we see a greater range of resistance than across all species. That's evolved very quickly. And it appears to have evolved independently. So when we look at the genetic similarity between populations, we see that geographically paired populations group out together. If resistance had evolved once and then spread, we would expect all these resistant populations to be genetically similar. And we don't see that. So we conclude that this resistance has evolved multiple times independently. So it's evolved a bunch of times. And it's evolved very recently, right? If you take sediment cores from these sites and you go back in time, these started accumulating in the 1950s, peaking in the 1970s or so. So we're talking a few dozen generations that this is evolved in. So it's evolved very quickly as well. So how do you get there? How do you get this super resistant animal from a really sensitive animal within a few dozen generations? So we started off by looking at what a resistant animal is doing differently at the molecular level, gene expression level, when you challenge them with these chemicals during development. So these chemicals are exquisitely toxic during development. They're teratogens. Okay? So we bring them in the lab for a couple generations. We expose them. We look at a transcriptome response. So these are data with that ancient technology of microarrays. And what we see for our sensitive site populations, we see this set of genes that gets turned on or transcriptionally activated with increasing doses of PCB126 as our model toxicant. And we know what these genes are. So we know a little bit, actually we know quite a bit about the molecular mechanism of action of these chemicals. So when things like dioxin or PCBs or PAHs get into the cell, they bind to the aryl hydrocarbon receptor. And this now ligand-activated transcription factor translocates the nucleus, forms a heterodimer with ARNT, and then binds to transcription factor binding sites and turns on this battery of genes. Transcriptionally activates this battery of genes. So these genes are these genes, genes that we expect to be turned on by this ligand-activated pathway. When you look at the resistant populations, even at this really high dose, the dose that's normally lethal, none of these genes are turning on. And it's not like they're blocking the chemical from coming into their body. Their body burdens are just as high. It's just this pathway is not turning on until you crank up the dose a couple of orders of magnitude, and then the pathway is turning on. Right? So we conclude that AHR signaling pathway appears to be desensitized in these tolerant populations. And this makes sort of mechanistic sense because you go to a mouse and you knock out AHR and you can't kill them with dioxin. So dioxins and PCBs, their toxicity is almost entirely mediated through activation of this signaling pathway. Okay, so that appears to be an important part of the mechanism anyways, at least for PCB126, one of the chemicals that are really contribute to the pollution of these sites. So what are the genes then? Okay, here's a phenotype. We see resistance. Here's a molecular phenotype. The signaling pathway is desensitized. But still, how do you get there at a genetic level? So we took sort of a brute force approach and basically looked for the genes that appear to be evolving under very recent and strong natural selection in these populations. So people had taken sort of the candidate gene approach over the last couple decades trying to figure out what was going on. But with genomic technologies becoming more accessible to those of us that work on NSF-size grants, this became possible to do in the Killifish. So we sequenced the genomes of 48 animals per population from these 8 different populations. And we basically scanned through these genomes looking for these needles in the haystack, regions of the genome that appear to be evolving by strong recent natural selection. And a lot of this work was done by Noah Reed, who was a postdoc in my lab. He's now faculty at UConn. And so just a quick cartoon of the kinds of signatures of selection that we're looking for. So say you've got a couple different populations here, okay? And within that population, you've got stretches of DNA where there's lots of genetic variation. So each circle is a SNP, a polymorphism, okay? So we see genetic variation within these populations. And then the environment changes, okay? And in this changed environment, this mutation is adaptive, okay? Here we've got a not polluted environment, so that mutation is neutral, okay? So after a few generations, this variant that provides a selective advantage increases in frequency, okay? So after a period of time, many generations of selection, that mutant is at high frequency compared to the reference population. But you see another thing. So that's one thing that we're looking for, big allele frequency differences that are bigger than sort of the genome-wide average of a polluted site population compared to their geographically nearby reference site population. Another thing you can look for as you're scanning through the genome are regions of reduced genetic diversity, because natural selection sort of seizes upon this variant, sweeps it to high frequency, but all the linked variation goes with it. So what you see end up with is a region of the genome that actually has reduced heterozygosity compared to your reference site population. So those are two different signatures of selection or signals of selection that you can look for in the data as you scan through. Okay? Signature 1 is allele frequency change and then reduced nucleotide diversity. And so this is maybe what it would look like as you sort of scan across a stretch of DNA. So here's maybe a few hundred kilobases, a stretch of a chromosome. So we see this increase in allele frequency change. FST is a measure of that. And then a reduction in nucleotide diversity in our resistant population compared to the sensitive population. Right? So that's what we're doing. We're scanning the genome for these signatures of selection. Okay? So here's a bit of a complicated figure. And this isn't going to, there's not a ton to learn from this, but this is for each pair of populations, what we're plotting here is our allele frequency divergence as we're scanning across the genome. And we've got a genetic map now. So this is 10,000 scaffolds before we were able to assemble the scaffolds into chromosomes. And we've got a genetic map now, but I haven't updated this slide yet. So this is 10,000 scaffolds. But what we're scanning for here are these peaks and divergence. Peak, 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 compared to the genome-wide average, okay? And so we see a bunch of these peaks. Your eye's probably been drawn to this one here, which is a huge peak in each of the populations, and it's a shared peak. Each of the populations has the same peak. So that's a question, how do you get there from here, and do you recruit similar types of genetic variation each time you evolve this kind of resistance? And at least at this locus, it seems to be shared. Here's our view, essentially, of this reduction in pi, this reduction in nucleotide diversity. So what we're looking for here are regions of the genome where there's a particularly negative number. And what I want to draw your attention to is the coloring here. So the coloring, if you're colored, if you're not gray, it means that you've got particularly low nucleotide diversity, that you've got a signature of selection somewhere in this region of the genome. If you're colored blue, it means that region of the genome is a signature of selection that's shared in at least these three northern populations. And if you're red, it means that you're a signature of selection that's found only within that one pair of populations, right? So the conclusion from what we're seeing here is that there's lots of red and not much blue. So most signatures of selection are population specific, and very few are shared. But the strongest signature of selection, the biggest outliers, are the ones that tend to be shared. Okay? So the inference being that the most important targets of selection for each population tend to be shared among populations. And perhaps some of this adaptive fine-tuning is population specific. Okay. So let's look under the hood at some of these regions. So here's that scaffold that I first drew your eye to, scaffold 98, 93, that doesn't matter. And right in the middle of that scaffold is aryl hydrocarbon receptor, not aryl hydrocarbon receptor, and this is a gene that everyone's been sequencing as a candidate gene for a while. And it's not that, it's aryl hydrocarbon receptor interacting protein, okay? About 10,000 studies in PubMed on what AHR does, about a dozen studies on what AIP does. But it appears that mutants of AIP affect the ability of ligand-activated AHR to translocate in the nucleus and activate expression. Okay? So So still focusing on this scaffold with AIP, remember here's the genome-wide average of what genetic similarity among populations looks like. It groups geographic pairs together. Right? If we look at just that scaffold that includes AIP, this is what genetic similarity among populations look like. That our 3 northern resistant populations all share pretty much the exact same haplotype. Again, it's a huge signature of selection in the south, in the red site, but a totally different haplotype. Okay? So big signature of selection in all 4 of our population pairs, but a different molecular variant is under selection in different populations. And this is a theme that I'm going to be repeating over and over again to make a point. So that's AIP. I'm going to draw your attention to our biggest shared signatures of selection. Okay? Another one is over here, and under the hood there is, well, it's AHR itself. And part of the reason that people had missed the variation in AHR is, at least in one of these populations, it's a deletion. It's pretty much wiped it out. Okay? So in this population, a deletion is swept to about 85% frequency. But in the north, again, signature of selection from the genome scan data, top ranking, signature of selection, but no deletion associated with that sweep. Okay? So again, signature of selection in all of them. It's top ranking. Here in the number is the ranking of the region of tens of thousands of regions. How strong is the signature of selection? Top ranking region, but a different molecular variant in different populations. Okay? So in Killifish, there's 2 AHRs that are right next to each other. The deletion in the red population clips off the last exon of 2A and the first few exons of 1A. And actually, our RNA-seq data shows that a chimeric transcript is expressed. We don't know if it makes a full-length transcript or whether it makes protein, but if it does, it has 2 ligand-binding domains, and it's weird Frankenstein protein. So we're not sure if the deletion is a disabling mutation or whether this has an acquired new function, this chimeric transcript. But anyways, strong signature selection, all populations. We've got convergence at the level of gene, but divergence at the level of the type of molecular variant that appears to be under selection there. So that's that. Another big outlier is one of the key transcriptional targets of this signaling pathway, cytochrome P450 1A1. And there it's a similar but different story. Here, a duplication has swept to very high frequency in the northern populations. Again, big signature of selection in the south, but not associated with any duplication. Right? So here's the copy number in these 8 different populations. So here are our sensitive site populations. They've got the normal 2 copies, right? Actually, some of our sensitive populations have a low frequency of a couple extra copies floating around. But in our northern resistant site populations, there's some individuals that have up to 6 additional copies of the cytochrome P450 appearing in tandem along the chromosome. And that's swept to pretty high frequency in those populations. Again, big signature of selection at this shared locus, but duplication in the north, but no evidence of a duplication that's swept in the south. Okay? So that's the other sort of big shared outlier. So each of our sort of top-ranking shared outliers tend to be components of this molecular signaling pathway. But even when we look at the population-specific, some of the variants that are private to a particular population pair, we keep seeing molecules of the signaling pathway getting dinged by Darwin, right? So here's a big outlier that's targeting ARNT. Another population-specific outlier, it's a different copy of ARNT. In that population, cytochrome, another one there, another one there, another one there. So almost all of the molecules in the signaling pathway appear to be targets of selection in each of these populations. So the AHR pathway appears to be a common target of natural selection, but there are different variants in different populations, especially between the northern and southern populations. So we see convergence at the level of phenotype, including molecular phenotype, but that convergent similarity starts to break down. It's still shared at the gene level, but what types of genetic variation are being recruited are different in different populations. So, so one thing that I talked about in the introduction was this idea that it's hard to evolve resistance to pollution because pollution is a complex mixture of chemicals. And when you've got a complex mixture of chemicals and perhaps many molecular mechanisms of action, you've got to solve a whole bunch of problems at the same time. So what's going on with these killifish? These are really polluted sites with really complex mixtures of chemicals. So they're mixtures of dioxins, PCBs, PAHs, that have different sources, different industries have contributed these chemicals to the environment, and they're all there in the sediment at pretty high levels, normally lethal levels. And that's true. What's interesting about this set of chemicals, though, even though they come from different sources, and there's big mixtures, lots of congener variation, what unites all of these chemicals is that they all exert their toxicity through this AHR signaling pathway. That's also true in the south, where they have complex mixture of chemicals. But again, that complex mixture of chemicals sort of seems to channel all their toxicity through this signaling pathway. So by modifying this one signaling pathway, you're able to sort of repeatedly achieve this resistance to a pretty complex suite of chemicals. But it's not as simple as that, because we know that there are chemicals in high abundance that don't exert toxicity. They're really toxic, but they don't exert their toxicity through this signaling pathway. For example, some of the low molecular weight polycyclic aromatic hydrocarbons that come from like creosote or oil, they exert their toxicity by disrupting potassium currents and developing cardiomyocytes. Okay? And that appears to be AHR independent. It's not dependent on AHR. But when we look at the delayed rectifier potassium protein that regulates this potassium current, we see a very strong signature of natural selection in this red population that's particularly polluted with these chemicals, but no signature there in the northern populations that's not particularly polluted with those chemicals. Similarly, in the northern populations are also polluted with a different class of PCBs that also don't exert toxicity through the AHR signaling pathway. We actually don't know a lot about their mechanism of action, but they're neurotoxic. We don't know that much about the molecular, so we don't know which of these genetic variants in these populations contribute to the resistance to this neurotoxicity. But the answer is there somewhere in our data. Somewhere in our data is an answer to what's the molecular mechanism of this class of chemicals. Because if you're able to identify the genes that contribute to variable sensitivity to these chemicals, all of a sudden you've discovered something about the molecular mechanism of action, right? So we're working on sort of mining these data to try not only give us insight into how animals evolve quickly to complex chemicals, but to also give some mechanistic insight into how these chemicals work. So even though AHR is really important, and the different components of AHR and genetic variation within those components is really important for this adaptive phenotype, it's not the whole story. Even though by pulling big levers associated with AHR, you're perhaps able to resolve a lot of the adaptive challenges, the fitness challenges posed by this complex suite of chemicals. It's not that simple. That the adaptive genotype is more complex because the chemical mixture is more complex than that. So I'd like to draw your attention to a news story that's just emerging from our lab, where we're working with another set of populations, a different species. This is a Gulf Coast distributed species. And fortunately for us, unfortunately for everything else, there are horribly polluted sites along the Gulf Coast as well, including the Houston Ship Channel. Okay, so here, City of Houston is right about here. Here is Galveston. So this is Galveston Bay. And in this study, in the first study, the Atlantic Coast study, we were looking at sort of a fairly coarse geographical level, where we had sort of 4 pairs of resistant, polluted site sensitive, reference site populations. Here we did sort of a different strategy, where we're sampling at high density along this gradient of pollution, going from fairly clean sites to really, really polluted sites as you get further and further up into the Houston Ship Channel. Okay, and so the cool color populations are the ones that are in the really clean sites, and as you get hotter, you're getting to more polluted. And the hottest, blackest hawk is, think of burned to a crisp. Okay, so These are really, really polluted sites. So again, thousands of times the concentration that's normally lethal. And there are killifish populations living there that are doing just fine. Okay? So here's your index of cardiac deformities when you're exposing these embryos during development to an increasing concentration of these chemicals. And as we can see, your level of resistance, your lack of sensitivity to these chemicals improves the further you are up in the ship channel. And so your level of resistance is actually scaling quite nicely with the level of pollution. So natural selection seems to be dialing in your sensitivity very specifically to just how polluted your environment is, including extremely polluted sites. So this was kind of interesting, and we're again scanning through the genome looking for these signatures of selection, and we're asking, are the adaptive variants or the adaptive genes sort of the same between sort of intermediately resistant populations? Are they the same genes that are under selection in super resistant populations, or what's going on? And the story is kind of boring at that level, that it appears to be the same gene. So here, of course, the signatures of selection, which are these red peaks, are a little bit weaker in the intermediate populations, the red ones, compared to the ones that are the super resistant ones, the black ones. But if you sort of blow up this axis and you sort of stand on one foot, you can kind of see that most of the big variants, these variants that are really showing these strongest signatures of selection, are shared across all populations. But we stumbled upon something quite surprising and entirely by accident a couple months ago, and you guys are the first ones to see the data. So we started looking at this locus here. Under this huge signature of selection was aryl hydrocarbon receptor. And we found a big deletion there that had swept to about 95% frequency in these populations and about 20% frequency in these intermediate populations. Okay? So we look at the genotype along this region of the genome and think of this as sort of a principal component analysis. We're looking at sort of genetic similarity among individuals across all these populations. And we see that individuals from these burned to a crisp sites all have a pretty similar haplotype, and it's very different from the haplotype that you find at the sensitive sites. And the heterozygotes sort of cluster somewhere in between. So this is, we're not able to phase, if we were able to phase the data, then you'd see two distinct clusters. But the reason these guys are individuals, or intermediate, is because they're the heterozygotes. But basically, I want to show you that these guys are really different from these guys genetically, so it's a huge divergence. That's not a big surprise. The big surprise came when we just happened to put the data together with the Atlantic coast species data. And there we found that the adaptive haplotype in these burned to a crisp sites came from the other species. That this haplotype that swept the high frequency, that carries the deletion, is actually the haplotype that came from Fundulus heteroclitus, the Atlantic coast species, thousands of miles away. So when we look at heteroclitus as the Atlantic coast species, and we know there's genetic divergence between the north and south clade, and here's the Gulf Coast species, the haplotype on which the deletion happens compared to the wild type, and we see that this is a very recent introgression from Fundulus heteroclitus from the southern clade. This happened very, very recently. So we've been working with Graham Coop's lab and a couple of very talented students in his lab, Siobhan Yer and Kristen Lee, to bring sort of some coalescent-based models to try and sort of model some of the time course over which this happened. And the conclusions from running a number of these models over the last couple of weeks, literally we had sort of the conference call and sort of swapping figures about 2 days ago, is that the age of the introgression appears to be sometime within the last 10 to 50 generations. So it's very recent. And the selection coefficient is off the chart, that it's about 0.9. So this adaptive haplotype appears to have introgressed recently from this Atlantic coast species. And when you consider the distance that must have traveled here, it's pretty huge, right? So here's Houston, and here's the closest population of the Atlantic coast species. They range down to northern Florida. And think of the huge tract of sort of poor habitat, at least salt marsh habitat, that separates these two, okay? This little dude isn't going to swim that far in 10 to 50 generations. So given what we know about the biology of this animal, they've got very high site fidelity. Their lifespan is usually lived out over the range of a couple hundred meters within a marsh. Mark recapture studies have shown us that this transfer, that this introgression almost certainly has to have been human mediated. Either through, you can speculate, either through ballast water of a ship. We know that fish are moved around through ballast water, or through, I don't know, maybe some seagrass restoration projects where they grab the sea. You can imagine a lot of different scenarios. We can't speculate too much, but it seems obvious to me anyways that this introduction, because it's so recent, almost certainly had to be human mediated. So the implication of that being that almost all invasion biologists, people that study invasive species, would agree that most introductions are deleterious for the native species that live there. But perhaps this is an example where the introduction, sometime in the last few decades, has actually contributed to the rescue of the Gulf Coast species, certainly from extirpation in these extremely polluted environments. So in summary, what we find for this vertebrate animal that's evolved resistance to these complex suite of chemicals, we see that this one signaling pathway is the repeated targeted natural selection. Okay, there's a really strong footprint of natural selection here. So, so the implication being that this pathway is a molecular or a mutation target that has a very large effect on phenotype. We go from sensitive to 8,000 times resistant very, very, very quickly. But we see that different molecular variants of AHR genes are recruited in different populations. So the implication is that there are a lot of molecular ways to pull this sort of common phenotypic lever. We also see that selection is targeting genes outside of the AHR pathway, genes that mediate different types of toxicity from different types of chemicals. So adaptations to pollution isn't simple because, well, pollution isn't simple. We see, at least for some of these AHR variants and some of these others, that they exist at some low frequency in the sensitive site populations. And you saw for at least that duplication at cytochrome P450, about, that was at a reasonable frequency. About 5% of animals in one of the reference site populations had at least a few extra copies. So that suggests that, standing variation, like theory would predict, large levels of standing genetic variation are going to be really important to enable very rapid adaptation. Basically, if you're facing a radically altered environment that's threatening not your relative fitness, but your absolute fitness, you have to be able to play the cards you have in your hand. You can't wait for new mutations to arise in these radically altered environments. And we see an example of adaptive integration from a geographically very distant population, happened very, very recently, suggesting that in some cases, perhaps, human-mediated introductions could contribute to a gene pool, expand the gene pool of native species, perhaps enabling evolutionary rescue in some scenarios. Okay? So the take-home from this, I wouldn't call this a paradox, but it's maybe an irony, is that these species that evolve very quickly to our poisons have huge population sizes, therefore huge reservoirs of genetic diversity. They have rapid generation times, okay? So a couple of years for them is very different on an evolutionary timescale than a couple of years for these guys when you're talking about the timescale of environmental change. And these guys are exposed to simple poisons with high specificity, whereas these critters here tend to have very small pools of genetic diversity from which they can draw very quickly, long generation times, and are exposed to mixtures. And so killifish, you might say, well, they're in here, what's going on? When you think of nucleotide diversity across taxa, of course, the arthropods have much higher levels of nucleotide diversity than vertebrates. But when you look at killifish, here they are. Compared to this other paper that was published in 2012, looking at a bunch of species, killifish have almost the highest levels of nucleotide diversity for any vertebrate species. They're essentially arthropods when it comes to nucleotide diversity. And here we are way down here. So genetic diversity is a crucially important part of this. So these are the people that actually do the work in the lab. I've got fortunate to have a very talented group of graduate students and postdocs. These are the people that I get to play in the sandbox with, which has been a really enriching experience over the last decade or so. And with that, I'd be happy to address any questions if there's time.